Hello everyone, today our group is going to present a science news story. The topic we have chosen is cells have more many organs than researchers thought and bound by membranes. These recall organisms challenge biology's fundamentals, and we are going to explain what the news is about. Think back to the basic biology class you took in high school. You probably learned about organelles, the little organs inside cells that form compartments with individual functions. For example, mitochondria produce energy, lysosomes recycle waste, and nucleus store DNA. Although each organelle has a different function, they are similar in that everyone is wrapped up in a membrane. Membrane-bound organelles were the textbook standard among scientists. Those cells were organized until they realized in the mid-2000s that some organelles don't need to be wrapped in a membrane. Since then, researchers have discovered many additional membrane organelles that have significantly changed how biologists think about the chemistry and origins of life. He was introduced to membrane organelles, formerly called biomolecular condensates, a few years ago, when students in his lab observed some unusual blobs in the cell nucleus. Unbeknownst to him, we had actually been studying biomolecular condensates for years. What he finally saw in those blobs opened his eye to a whole new world of cell biology.
Light a lava lamp to get a sense of what a biomolecular condensate looks like. Imagine a lava lamp as the blobs of wax inside fuse together, break apart and fuse again. Condensates form in much the same way, though they are not made of wax, instead a cluster of protein and genetic material, specifically RNA molecules in a cell condenses into gel-like droplets. Some protein and RNAs do this because they preferentially interact with each other instead of their surrounding environment, very much like how wax blobs in a lava lamp mix with each other but not the surrounding liquid. This condensate creates a new microenvironment that attracts additional protein and RNA molecules, so forming a unique biochemical compartment within cells. As of 2022, research has found about 30 kinds of these membrane biomolecule condensates. In comparison, there are around a dozen non-traditional membrane-bound organelles. Although easy to identify once you know what you are looking for, it's difficult to figure out what biomolecular condensates exactly do. Some have well-defined roles, such as form reproductive cells, stress granules, and protein-making ribosomes. However, many others don't have clear function. Non-membrane-bound organelles could have more numerous and diverse functions than their membrane-bound counterparts. Learning about this unknown function is affecting scientists' fundamental understanding of how cells working.
This topic is about protein structure and function, and this is a new article related to it. Traditionally, scientists thought a protein shape determined its function and a fixed 3D structure was essential. But new research shows that many proteins form biomolecular condensates made from intrinsically disordered protein or IDP. IDP have a non-stable shape, yet they still perform specific jobs. Their function depends on the environment and how they assemble, not just a rigid structure. This is important for three reasons. One is challenge the old rule that shape equals function. Two is help explain disease like Alzheimer and ALS. 3. It gives clue about the origin of life, showing early life could work without complex folding. In short, ATP and biomolecular condensates show that proteins are more flexible and versatile than we once thought. They open a new world in biology and help us understand cells in ways we never imagined.
Bacterial cell. Researchers have also detected biomolecular condensates in prokaryotic or bacterial cells which traditionally were defined as not containing organelles. These findings could have profound effect on how scientists understand the biology of prokaryotic cells. Only about 6% of bacterial proteins have this orderic region lacking structure, compared with 30% to 40% of eukaryotic or non-bacterial protein. But scientists have found several biomolecular condensates in prokaryotic cells that are involved a variety of cellular functions, including making and breaking down RNA. The presence of biomolecular condensates in bacterial cells means that these microbes aren't simple bags of protein and nucleic acid, but are actually more complex than previously recognized.
The next topic, I'm gonna talk about the origins of life. Biomolecular condensates are also changing how scientists think about the origins of life on Earth. There is ample evidence that nucleotides, the building blocks of RNA and DNA, can very possibly be made from common chemicals like hydrogen, cyanide, and water. In the presence of common energy sources like ultraviolet light or high temperatures, on universally common minerals like silica and iron clay. There is also evidence that individual nucleotides can spontaneously assemble into chains to make RNA. This is a crucial step in the RNA world hypothesis, which postulates that the first life form on Earth were strands of RNAs. A major question is how the RNA molecules might have evolved mechanisms to replicate themselves or organelles into a protocell, because all know life encloses in membranes. Researchers studying the origin of life have mostly assumed that membranes would also need to encapsulate these RNAs. This will require synthesizing the lipids or fats that make up membranes. However, the minerals needed to make repeat likely were not present on early Earth. With the discovery that RNA can spontaneously form biomolecular condensates, lipids wouldn't be needed to form protocells. If RNAs were able to integrate into biomolecular condensates on their own, it becomes even more possible that living molecules are lost from non-living chemicals on Earth. This is an interval. New treatments. For me and other scientists studying biomolecular condensates, it is exciting to dream of how this rule-breaking entities will change our perspective on how biology works. Condensates are already changing how we think about human diseases like Alzheimer's, Huntington, and Lou Gehrig. To this end, researchers have developed several new approaches to manipulate condensates for medical purposes, like new drugs that can promote or dissolve condensates. Whether this new approach, the treating disease will bear fruit, remains to be determined. In the long term, I would not be surprised if each biomolecular condensate is eventually assigned a particular function. If this happens, you can bet that high school biology students will have even more to learn or complain about in their introductory biology classes.